which is Dr. Vineet. And Professor Vineet, hi. So,
good afternoon.
Hi. Good afternoon. So, Professor Vineet is an associate professor in ICER Bhopal. He has a PhD in bioinformatics from IGIB, which is my alma mater. And while going through his degrees, I was very interestingly curious to see a master's degree in intellectual property law. He also has a master's in that. And his lab works on metagenomics, genome, system biology, machine learning, using machine learning techniques. And he has some really cool tools named like MP3, MP4, which are about finding pathogenic proteins using metagenomic data and genomic data. So we look forward to actually hearing his talk about how he utilizes genomic and metagenomic data in his research. So over to you, Professor.
Yes, thank you very much, and for the nice introduction. So I'll go to my slides and then I'll introduce myself there, right? So, okay, share screen. Okay, it's visible to all?
Yes, it's visible.
Yes, good afternoon. I'm Vineet Sharma. My lab works in the area of metagenomics, and we have done several. So we are the leading researcher in human microbiome in India, and we have done some excellent new novel eukaryotic genomes, which I'll present to you. And also I'll show a glimpse of the AI-based tools that we have developed using this microbiome data. So our human microbiome is rightly to be called as a second genome because we have more number of bacterial cells than compared to the human cells, and they are all over our body. And in fact, if you see that in the gut, we have almost 2 kilograms of microbes, more than 1,000 species. And like, for example, when you say that I have a gut feeling, are actually, talking about the feeling of the microbes that are there in your gut, right? So to find this genetic information, which is there on our skin or our scalp or our gut or the human microbiome, we have to employ metagenomics, where we directly go for sequencing without any culturing. So we have explored the human microbiome from Scalp. That was done with L'Oreal with over 1,000 samples. We have looked at the gut microbiome of the autistic children and find some novel markers. We explored the gut microbiome of the tuberculosis patients before and after they were treated with drugs. We explored the oral microbiome and in association with colon cancer. We published the largest gut microbiome from India so far 2 times. Right, one paper recently came a week ago in Nature Biofilms and Microbiomes, where we have more than 200 samples. Then we also looked at how a condition like colorectal cancer can happen, what bacteria are responsible for that in our gut, and I'll also give a glimpse of that today. We are also looking at vitiligo and other skin, metagenome and metabolome. And I will present a tool where we have shown that how drugs that we take, right, are metabolized by the human gut microbiome and may lead to their inefficacy and like that. So coming to the human gut microbiome, we have taken samples from all over India. Not exactly all over India, but like the published one is from 6 places, but others are in progress. So India shows a lot of variations in food, dietary habits, and geographical locations in terms of water, other things, that climate. Then there are other factors, confounding factors such as age, vegetarian, non-vegetarian habits, right? So, so we have to, consider all these factors. So for this, an Indian gut microbiome was never explored before in terms of metagenome. Only some glimpses of like 16S-based studies were available. So first of all, we constructed the Indian gut microbial gene catalog to see that what function the Indian gut microbiome has and compared with the world gene catalog. And we were happy to contribute almost 1.5 million new genes to the World Gene Catalog. That means that is unique to India. That is contributed by the Indian gut microbiome. So we established the gut microbiome. We also established the Indian gut metabolome, which was never explored before. We looked at the function of this gut microbiome in health. I will present that later in my slides. And particularly, the human gut microbiome in India was rich in carbohydrate and energy metabolism functions, right? And for this, we employed both 16S shotgun and metabolomic approaches, and also did the serum metabolomics to validate the findings that we find in the gut. Right, so if you look here, you can see that the Indian gut microbiome looks very distinct from the other gut microbiomes, right? And particularly, if you see that, they cluster very separate and they have a very large, you can say, inter-sample distances also because of the variation that we have in the diet. But despite of these variations, we cluster together. So that means there is something which is common across the Indian geographical regions, and but still we are very different from the other populations. And particularly the bacteria like Prevotella, Megasphaera, Lactobacillus, etc., were distinct in the Indian gut microbiome. And the gut microbiome was enriched in the carbohydrate metabolism genes, right? Which was a very distinct property. So we take a lot of dietary fibers. We use a lot of fibrous. Even our wheat that we use is fibrous, and so it is our daily intake. So we have the microbiome that fits to that kind of food that we take. And so, for example, this Prevotella. So in India, it is almost 70%, right? It can go up to 70%, 30 to 70%. But whenever this Prevotella copri goes beyond 5% in other Western population, they suffer from all kinds of inflammation and other problems, right? So that I'll come to this thing later, right? So in terms of enterotypes, it's a concept that you try to see that, okay, what different type of bacterial genera dominates in a population and what are the predominant ones. So there are 3 types so far, driven by Prevotella, Bacteroides, and Ruminococcus. So the Indian gut microbiome fits the enterotype 2 that is driven by Prevotella. So we found that. And coming to the Prevotella copri, that is the most abundant bacteria in the human gut, we see that it has all the genes and pathways for the branch chain amino acids synthesis, both basically branch and non-branch chain amino acids. And looking at the different location that we had, we could see we propose 2 kinds of models, right? One is there could be a situation like in location 1, that was say the northern India. The bacteria mostly has these branch chain amino acids biosynthesis genes, okay, which is predominantly present in Prevotella and other species, right? So when this is the situation, this gut microbiome these microbial cells do not have the transporter for BCA from outside because they are synthesizing on their own, and that is why they also do not produce. They do not take it inside. So this is a microbial cell, for example. So they won't take it inside, and that is why they are also not detected in our feces. So our fecal metabolome does not contain these branch chain amino acids. But our serum metabolome, when we looked at, they contain these branch chain amino acids. That means it is absorbed by the human body. So microbes produce and it is absorbed by the host. But in the other case, bacteria do not have. So when they are devoid of Prevotella, they do not have these BCA biosynthesis genes. And in that case, the bacterial cells were found to have the transporters for BCA, like branch chain amino acids, inward transport. And the bacteria accumulates this, and these are detected in our feces, right? Because we are doing the bacterial metabolome from the feces, right? So this shows that the dynamics of BCA, and this is important because branch chain amino acids are linked to diabetes and other hypertension, these kind of situations. So if they are more in the serum, then we are more exposed to these kind of cardiovascular and diabetes kind of risks, right? This is the last week that is published in Nature Biofilms and Microbiomes. I could not prepare much slides from this, but the message from here is that we explored Prevotella in a lot more detail across the globe. We considered 9 populations here, did comprehensive analysis, including both Western and non-Western populations. And we observed that, and basically this was also the largest gut metagenome so far that was examined here from India. So we noted that, of course, Prevotella copri is the most dominant one. We constructed, and basically it is also less, or you can say understudied bacteria, because it is dominant in this part of the world, but not in the other part. Okay. So we had to, to do more comprehensive work on Prevotella. We had to construct a database also. So we constructed the genome as well as the gene catalog for the Prevotella. And then we looked at the polysaccharide utilizing ability of this microbe. And we found that this has a lot of PULs, polysaccharide utilizing loci on their genomes, which have a diverse array of enzymes which can metabolize the different kinds of fiber-rich diet that we take. So this proves that how the fiber-rich diet that we take, that we consume, is metabolized, and these and on the other side, when we looked at the Western gut microbiome, we observed that they did not have this bacteria, but the Prevotella that they have, the Prevotella genomes that they have, the other species, are mainly from the oral origin. That means, or that is the mouth to gut axis theory, right? And those species are also associated with the gut inflammations such as ulcerative colitis, irritable bowel syndrome, or even colorectal cancer, right? So these findings and the roles of Prevotella and their presence and absence in the 2 parts of the world shows and provides more insights into their roles in metabolism and disease, and also provide leads for having new kinds of prebiotics and probiotics which can remove this kind of dysbiosis from the gut, because so far there is no gram-negative bacteria-based probiotic which is available, right? Which has to be there for the Indian gut microbiome. And basically, not this latest work, but the earlier works were very nicely covered all by the media also, right? I'm just giving you glimpses of that. So then we explored, okay, so we looked healthy disease gut microbiome established, and that gives us an opportunity to explore the colorectal cancer and other, gut disease and gut conditions, right? So colorectal cancer is the 3rd most abundant cancer in the world and the 2nd leading cause of death. But in India, its incidence is low because we take a lot of flavonoids here, right? And but the adoption of Western lifestyle is disturbing the gut microbiome, which we also observe in a few cases. And we still cannot detect, colorectal cancer by non-invasive methods. So what we did here, we examined the colorectal cancer microbiome samples from the patients that had colorectal cancer in different stages, 1 to 4, right? And compared with the Hadley related gut microbiomes, right? And here what we found is that the colorectal cancer patients' gut microbiome was really deficient in pathways related to biosynthesis of several essential amino acids, which shows that they might have a deficit deficit contribution from the gut microbiome. And when compared with other gut microbiomes, also with the CRC gut microbiomes, we could find the markers that clearly distinguish the colorectal cancer from the healthy gut microbiome, right? And we can use these markers to develop diagnostic kits, which we are also developing. Right now, we have several leads for that. And this can find applications in non-invasive diagnosis of and early diagnosis of colorectal cancer, even in the first stage, just by looking at the gut microbiome and its metabolites. And this work was published in M Systems. So why, and among these findings, we found flavonofactor plauti, and we established its association with colorectal cancer. So far, its role was not known in colorectal cancer, but we established its association, and it was very distinct from the healthy gut microbiome also. And how, basically it promotes or associated with colorectal cancer is because it has the ability to degrade these flavonoids, right? And flavonoids have a protective role in colorectal cancer, right? Or to protect against cancerous conditions in the gut, right? So this, the presence of this microbe, it can degrade the flavonoids and even their serines of the flavonoid molecules, and basically, and we establish this not by the metagenome, by the bacteria, but also from the fecal metabolomics, right? So basically here we found a new bacteria which can be associated with colorectal cancer, which was not known before. And this was also widely covered by various media and reports, right? Okay, then there is another condition like in autistic children. The autistic children gut microbiomes were reported from other countries, but not from India. And typically in those countries, the Western countries, right, they take diet which is gluten-free. But in India, we take diet, even this autistic children takes diet which, they may contain gluten, and there is no such, restriction like that, right? So what this comparison of the autistic gut microbiome of the gut microbiome of the autistic children and the healthy children, we observed that lactobacillus particularly was almost 1,000 times in the autistic children, right? And this finding we also confirmed in the other US population, autistic children data, which was taking gluten-free diet. So both in the gluten-free and with gluten diet, we could find the same findings that this bacteria was almost 1,000 times enriched, right? So this, which could be related to the types of gastrointestinal problem that these children suffer, and again leads, provide more leads for new types of probiotics which can be used here, right, which can take care of this gut dysbiosis. Okay, then earlier with collaboration with IGIV, we also looked at the gut microbiome of the tuberculosis patients, and so we took samples from different stages, the healthy samples and the patients, tuberculosis patient which never had any antibiotic, that is the start day, and then after the antibiotics followed them, and we observed that, okay, first we showed that, okay, antibiotics are working and the amount of mycobacterium in the gut is decreasing, right? Because from the sputum we ingest bacteria in the gut, right? So it is not that this bacteria is a permanent resident of the gut, right? But through the sputum it is there. But when we take antibiotics, we could see that its number is decreasing, and we could see that the gut microbiome of the tuberculosis patients were deprived of propionate and other short chain fatty acids producers, right? Which means a reduced immunity. So we don't know cause or effect thing, but it could be that the bacteria is actually, MTB is actually manipulating the gut microbiome through that, or it is the reduced immunity which is leading to this dysbiosis. So this still has a lot of scope for further research, right? Then coming to the skin microbiome, see, skin is the largest exposed organ of the human body, and the conditions in India, the heat, the dust, the cold, right? And right, so this shows that there could be a lot of variations in skin, scalp, and right, and this was never explored before. And basically this has to be looked in both genders, different age groups, because skin shows a lot of aging effects, right? And both metagenome and metabolome both are important here. So we did the largest scalp microbiome study in the world so far in association with L'Oreal, and we looked at the healthy and the dental scalp microbiome, right? The cohort was a large cohort, more than 1,000 samples were there, right? And typically it was known that from 16S-based studies, amplicon-based studies, that there are two main players in bacteria. We looked at both bacterial and fungal microbiome. The bacterial players were Propionibacterium acne and Staphylococcus epidermis and epidermidis, and two Malassezia strains, Globosa and Resicta, right, which are associated with dandruff, right? And in fact, it is their proportion that matters that we showed here. You may have both kind of these species, but their proportion leads to both healthy and dandruff conditions, right? So we carried out the study using Amplicon, Metagenome, ITS sequencing for the fungal and their metagenomic sequencing also, and looked at the role of both fungal and bacterial microbiomes in what benefit they provide to the host in the healthy state, and how this healthy state is disturbed in the case of dandruff. So for these four players that were there in all the healthy and dandruff scalps, you can see that their proportion varies a lot across individuals. So each bar here is one individual, and you can see that the proportion of these major players shown by these four colors, 6 colors, because there were 40% of fungal species on our scalp were unknown. So, you know, they had to be accounted for here. You can see that it varies a lot, and right, in terms of healthy and dandruff, if you compare, you know, you see a very different, landscape, whereas in the healthy and dandruff, in case of bacteria also, you see very different landscape. The colors show that, right? So which shows that, and these colors are present in all. That means it is present across healthy and dandruff, but the proportion varies. Now, what varies here is, when we looked at these four species, we see that the proportion of this Propionibacterium acne, which is supposed to be associated with the healthy scalp, versus Staphylococcus epidermis. Okay, so their proportion varies in the healthy and dandruff scalps, whereas the healthy associated M. globosa was more compared to the Resicta in case of the dandruff scalps. So in case of both dandruff and bacteria, we identified that, you know, how this dysbiosis and what percentages are leading to this kind of dysbiosis. And then we examined their functions and found that this healthy scalp microbiome for the fungal associated with dandruff is actually enriched in functions that helps the fungus to survive on the scalp, right? Like lichen synthesis, which is important for the attachment of the fungi to the scalp and like that, right? And it was mainly contributed by Malassezia resicta, which was found to be more associated with the dandruff fungi. But in case of bacteria, the healthy scalp bacteria were contributing a lot of important things to the host, such as they were doing the biosynthesis of several vitamins and amino acids, okay, which can be absorbed by the host. We also could find the transporters for these. So we could look at the transporters of these biotin and other metabolites which are produced by the bacteria, whether the host can absorb it, and we could find those transporters that the bacteria can throw it out. And there is a well-known mechanism by which the scalp can absorb the nutrients from outside. Right. So which shows that this bacteria on our scalp in the healthy state has their roles and importance for the host. Right. And along with this, we also looked at the host factors, such as the itching, total dandruff score, sebum content, hydration, right? And again, the species that we showed that were associated with dandruff, such as S. epidermis and the uncultured Malassezia and the Malassezia resicta, they showed positive association with the dandruff parameters, whereas the species such as Propionibacterium and Malassezia globosa in fungi and bacteria respectively, they showed negative association with these dandruff parameters. So this was also clinically validated. And so basically, this is the model that it proves that basically this particular bacteria, Propionibacterium, contains the genes for the biotin biosynthesis. Also had the transporters for the export to the host, right? And along with that, the other nutrients, and they are absorbed by the scalp in case of the healthy conditions. And when this microbiome is disturbed, it leads to the dandruff and other, scalp problems. Then the other part of the story was whether the coconut oil that is widely used in India, whether that has some effect on the scalp, right? And for this we use a typical neutral shampoo as the control, right? And the same set of individuals that were there that we examined earlier were given these kind of treatments over a period of time, right? And we looked at during the treatment after relapse phase that how this whether the coconut oil is causing any benefit to the host and to the scalp, and how this benefit is reflected in terms of the microbiome, right? So we observed that the fungal and bacterial species that we found to be associated with healthy were promoted, particularly the bacterial one. Fungal did not show like too much change, right? So the healthy bacterium like Propionibacterium showed an increase, whereas in case of dandruff also, which had lower Propionibacterium earlier, also showed an increase of Propionibacterium after the treatment, right? Which was also observed in the relapse phase, right? So, and we also looked at how this is happening in terms of pathways. So I'll not go into details of that, right? So which showed that coconut oil provides beneficial effect to the host, and it promotes the healthy bacterial species which are associated with various amino acids and vitamin biosynthesis on the scalp. So these were the largest scalp microbiome studies so far, and they revealed that the Indian scalp microbiome has a distinct composition of Malassezia, which is still unknown. Almost 40% is, we don't know yet what is that. So the anti-dandruff therapies that we use may not be able to target our dandruff to the greatest efficiency, right? And we showed the role of bacterial microbiome in the scalp homeostasis and the nutrient conditions, right? And this understanding will have a lot of applications in anti-dandruff therapies and maintaining the healthy scalp, right? And this was also widely covered and cited by the different media, right? Then I'll take you to now how we use this genomic data in AI, right? Using AI ML approaches, right? So basically this, I'll start with ML and then I move to the AI, right? In case of, so we developed two things. So we looked at skin and gut. So we developed DrugBug and Skinbug, two tools, right? So how it works is, so what is the problem and why it is needed and like that I'll show. So basically we take oral drugs that goes to your gut and it sees like millions of microbes that are there, right? And these microbes may contain enzymes which can do the promiscuous metabolism of the drug molecule, because the structure of the drugs may look similar to the structure of their own substrates, right? And like non-specificity of promiscuous metabolism is one of the characteristics of enzymes, right? So which leads to sometimes this activity leads to the activation of drug, or can make an active drug inactive or basically less effective, or it can also lead to toxicity by the drug, right? And that is why we have responders to a drug and sometimes non-responders, and also sometimes it shows toxic reactions. And the famous example is the cardiac drug digoxin, for which, and this also tells us how difficult this problem is. Basically, 2 science papers in a span of 20 years, right, could find the first science paper found the bacteria the bacteria which can degrade the drug digoxin, which is a cardiac glycoside, right? And the second paper after a span of 20 years could show that how this works. Right, so basically this is an important challenging problem which should be addressed by the data that we have, right? And that is why we used machine learning approaches here. So what we wanted to find is the exact microbe, the metabolic enzyme that is doing this promiscuous metabolism, the actual site of metabolism, the population specific responses, and what happens after the drug is broken down, whether it is still effective or not, like that, right? So if you see here, this slide shows that how, basically what could be the consequences and what happens in the gut. Basically, microbes can do a reduction, hydrolysis, and of the drug, which are the most common ones, right? Oxidoreductases, right? Sometimes the presence of microbes leads to a wanted response of the drug. Sometimes it's deactivate, like a germ-free and colonized animal that you see here, and sometimes it leads to toxicity, right? So microbes do have a important role to play in the gut when we consume drugs, right? And so we developed a tool called DrugBug, which is based on machine learning approaches, and it could find the microbe, the enzyme, and the site of action where these enzymes were actually attacking. And we also showed and validated these predictions by real examples, the known examples, and also by molecular dynamics approaches, right? Okay, so I'll show a common slide for skin and the gut microbiome. So basically, this single microbe contains its own set of interaction that are happening, right? There are with different enzymes which are acting, it has its own interactome, right? But when we see in terms of like skin and scalp, which contains millions of these microbes, what we are dealing with something like this, right? So billions of interactions which are happening all the time, and we have to see it is not that one enzyme will act at once, right? It could be there could be a substrate, one enzyme will act, the product can again be a substrate for the next enzyme, right? And this way there could be a cascade effect, right? So this was also one of the critical things here. So how it works is like for any input molecule, it converts that into features, which are descriptors and fingerprints. And we use the random forest and deep learning methods, right, to predict the enzyme in 3 steps, I will say. First, it predicts the broad class of that enzyme. Then it predicts like that is 1 to 6 classes among these 6. Then it looks at the subclass. And after that, we use molecular similarity and by Tanimoto coefficient and the KNN approaches to look to find the exact enzyme which is doing its job, right? And then we map it back to what possible microbes contain this enzyme, right? And this way, and all the reactions that are possible to that enzyme, and this way we could see that how this is that enzyme microbe combination is actually leading to this promiscuous metabolism. Okay, so why it is important is like if we know the site of metabolism, that okay, I show that okay, this enzyme of this microbe can say reduce this bond in this drug, right? So, and drug manufacture is a very costly process, right? And its clinical validations and trials and etc. Right. So if we know this information beforehand, we can very well protect this bond or the site where this microbe is acting and make the drug universal so that it does not show population-specific variability, right? And also, once the drug is already there in the market, if we know this information, we can protect this drug. I'll show some examples here, right? And we also do like metabolomics to confirm our findings, right? So like this drug desoxin, again, I'll take this example. So we know that, okay, so this is the drug structure. It has a glycon and aglycon moiety, and this is the active moiety here, right? So population A is a like typical microbiome, which is there in healthy, right? But in population B, they have contained this microbe agar-thala, which can reduce this bond, right? So basically, this reduces this bond, and this structure now becomes inactive. Okay, so now this drug cannot function. Okay, so this information was known. So what DrugBug could find is it could find not only the microbe that was known, Agar-thala lenta, but also other microbes and the enzyme which can also do the same kind of reaction to this drug molecules, right? And for some typical cases such as desoxin and amphetamine, we also showed that how this drug, whether the drug that we propose to, the enzyme which is proposed to act on the drug is actually binding to that. So we showed by molecular dynamics that, okay, yes, it is binding to that drug. However, it is binding with much lesser, strength. So in case of natural substrate, which binds with 5 hydrogen bonds. This that is fumarate. This digoxin, which is the promiscuous substrate to that enzyme, is binding only with 3 bonds, which shows that basically this is not so strong bond. Okay, but still it can do this metabolism, right? So here in this case we propose that okay, if you provide fumarate along with the digoxin, so basically which is a natural competitor to the digoxin, right? So it can again lead to the same kind of efficacy for that drug, right? And similarly for amphetamine, we showed that amphetamine, yes, it binds to the enzyme that we predicted, and we also showed with the metabolomics that okay, this presence of this enzyme is actually leading to this so basically this part is still in progress, right? That how this is deactivating the drug and what are the resultant molecules by mass spec. So this is the server that we constructed, DrugBug, and you can submit or a molecule of interest by SDF format, right? Or right, and basically it or you can select different models, that is hybrid or fingerprint or so upsampling is one thing here, because whether you want to upsample the enzymes which are there, because EC1 to 6 show a lot of variation in the numbers that they have. So in DrugBug, basically the initial version we had this option, but later on we could take care of this option in the Skinbug, where instead of upsampling we did downsampling, right? So, and it will tell you the exact enzymes and the pathway, right, and the reactions by which it is metabolizing, right? I'm not showing the example here, but I'll take you more through when I go through the next version of this, that is the Skinbug, which is recent and published in iScience this year. So similar situation happens on our skin. So there could be a lotion or a deodorant which is supposed to work on the skin, but maybe the population have different skin microbiome, and it may not have that kind of lasting effect, or even leads to itching and other kind of reactions on the skin. Okay, so but can be predicted before, right? So we developed this tool called Skinbug, okay, using almost similar strategy as DrugBug, but with much enhanced features, and also using deep learning as one of the models along with the random forest. So combination of both AI/ML, right? So and here in DrugBug we could not predict this cascade, that okay, the starting molecule could be this, then one enzyme acts on it, then this is the product, then the product is again the substrate of the next enzyme, because skin is having so many enzymes and gut is also having so many enzymes. That we could not achieve in DrugBug, but in Skinbug we could find the cascade of metabolic reaction, the exact reaction pairs, the reaction centers where the reaction will be happening, the microbial enzyme will be acting on the structure, what products it will form, and whether those products are toxic or not to the host, right? Through the toxin, right, which is another tool that we developed, right? And we will also validate this with gene knockout and other metabolome-based applications. So first of all, when we looked at the skin metabolome, right, we could see that it shows a lot of variation in the different skin sites in terms of the 6 enzyme classes, right? And so this shows like, and you can see that only 10% of the total enzymatic reactions are common across all the 19 skin sites that we considered. That means we have to consider the skin site when we are making this kind of prediction. That okay, at this site this enzyme or microbe could be present, but not at the other site, right? So this is one of the things that that should be considered while addressing this kind of problem, right? And then by looking at the data and by showing with the normal PC and those based approaches, we showed that basically this clustering based approach will not work here because of the complexity of the data, and it does not explain the variability in the data. So we have to employ, this gives more opportunity for machine learning and deep learning kind of methods to address this complicated issue, right? So we developed this tool, Skinbug, which can predict the exact skin microbe, the skin side, metabolic enzyme, and what kind of bias transformation is happening the metabolic reaction, the reaction center, and the resultant metabolites, and the toxicity, right? And we also validated it on a set of about 50 to 100, I think 50 we reported in the paper, xenobiotics, and the environmental pollutant that we are exposed to every day, which is actually called our skin exposome, right? So we used the different features, we derive features from these molecules or substrates, right? And fingerprints, both linear and circular, and the molecular descriptors were used for the training, right? So basically it goes like this. The input molecule, we calculate the molecular features and do the feature selection. We consider all kind of like linear, circular, and molecular descriptors, and then do the feature selection for the best optimal performance. And at the first step, we use machine learning as well as the neural networks to predict the first. So we did this independently, and then use the, if there is a consensus, we use the result further and explore this class. So first we predicted the 6 enzyme classes, and we looked at that, okay, what is the subclass, and then what is the exact enzyme, which is done in the module 3, right? Where for finding the exact enzyme, we use the molecular similarity by Open Babel and 10-meter coefficient of more than 95 value, right? And using KNN approaches to pinpoint the exact enzyme which will do this job. And then we, using the RDM pattern database, we looked at the exact reaction center that where that enzyme will act, so that we can predict that what are the resultant metabolites after that breaking down of that molecule, right? And then we also constructed the enzyme, metabolic enzyme species and skin side databases for to complement this information. And we could predict all possible reaction, the reaction centers, and enzyme and the microbe which can potentially metabolize anything that we apply on the skin or use on the skin or we are exposed to during our daily lives. And this is available here, metagenomics.icb.ac.in/Skinbug. And for the toxicity prediction, we developed another tool called Toxin. I'll not go to the details of this, but basically it's a, it employs the same machine learning methods within an forest, and right, we evaluate all the methods, and the method that works best, we use it further, and basically it shows very good accuracy on both real and test data sets, and it was also published earlier, right? So this is Skinbug. So you can upload your file or enter the PubMed ID, PubChem ID of your molecule, and then so for example this, and then it tells you that okay, this belongs to oxidative. The exact enzyme is this one, okay, but it basically this is the broader molecular class. This could be important because sometimes you are interested in what class of enzymes can act on this molecule to to be more comprehensive, right, or broad, right? And then the exact reaction centers where those enzymes are likely to act, right? And using that RDM. And we are in the process of constructing a module which can predict the resultant molecules also, so that we can also tell the toxicity of, toxicity predictions can also be made on these molecules, right? And the source of these enzymes and the microbes and their sites, the skin sites from where these microbes could be, so that for any molecule you can know beforehand that, okay, this skin site, this population, okay, may have an enzyme that can, reduce the impact of this molecule or can lead to some kind of toxic or, any unwanted reaction. Right, we validated on several molecules, but due to lack of time, I think I should move a little faster. So basically, we showed in our paper that for all the common molecules for which information was known or unknown, we could predict, in case of the known information, we could predict what was known, okay, and even more than that. And in case of unknown, we showed for the first time that, okay, this enzyme and these microbes can act on this. Right, the typical cases of benzopyrene, which we are exposed to every day, our skin microbiome, right, and the azodies. So here, like the predicted metabolism was this, and we could also predict, the same enzyme and the bacteria which can do that reaction. Okay, so for both the examples, and this was also widely covered. Okay, now coming to the last part, our other interest is genomics. So we want to analyze the genomes for the first time, the sequence and analysis. So we've published the first, we did the first genome sequencing analysis of peacock in the world. It is also the national bird of India, and it is very fascinating bird. It has, such nice feathers and trains, and Darwin was also perplexed looking at this bird because it still survived so long because it cannot fly much, but still, and it is very prone to the predators also, but still it could survive. And then what is the mystery of behind this, the typical colors that we see in the, feathers of peacock? So we did the first genome sequencing of peacock and compared with the other birds in, related birds. And we could observe two things. One, we explored the feathers, right? What is like whether there is some kind of evolutionary features in the feather genes. So we could find, okay, a list of genes that are highly evolved in terms of the feathers of peacock, right? And second, we could find a very strong evolution or a directed evolution in the immune related genes, right? So for this particular bird, basically there is a natural selection that the female peacock finds the mate looking at the eye spots. So basically it counts the eye spots in the peacock. And basically these eye spots are a true reflection of or indicator of the how strong the immunity of that peacock. Okay, and this way its immune system became evolved, right? So we could show this, and also did another followed it up with another study where we looked at the how sexual selection happens in peacock. So we published 2 papers out of it, right? So this was also widely covered, and basically our own Isagopal BSMS students contributed to this work, right? Okay, then we did the first tiger genome sequencing in India. The tiger genome sequencing was already done by Chinese, right? But we noted that their sequence quality was not good, right? It had a lot of errors. So we did the first tiger genome sequencing in India, and then we compared and we developed an algorithm called Skinbug, which could correct the assembly problems because there were several errors in the assembly, and we could correct, almost 1 million bases in the genome and almost 11,000 bases in the coding region, which is important because when we do the evolutionary study, we compare the coding regions mostly, right, to see that how the functions are evolved, right? So if the errors are there in the coding regions and in transcripts, basically it will lead to false results, right? So by the correct, so we did the sequencing, corrected the genome, produced a better genomic assembly, and then when we looked at the tiger genome, we could find that the genes that are involved in development and neuronal functions are very much evolved and in notch pathways. So basically, this tiger is can respond to stimuli. It can hunt and it has those kind of skills. So basically, this is very important. Okay, then coming to maybe the I think my time issue or something. Okay, I'll rush through. Then we did the first turmeric sequencing in the world, which is published last week in Nature Communications Biology. So far, the turmeric genome was not known, and we could confirm its diploid state. Did the hybrid sequencing both using 10x and nanopore, right? And produce a very, very high quality genome and also the transcriptome, okay? And showed that the origin of these genes, the curcuminoid genes, which are typical features of curcumin genome, right? Their gene structure, their evolutionary origin, and their adaptation in this plant, and also showed that how sorry, this is I should take it down. Yeah, and how this like evolutionary analysis, is important for the medicinal properties. So basically the message here is that this plant produces a lot of secondary metabolites which are of medicinal importance, but basically this is producing it for its own survival, for its adaptation to the environment, right? And typically this curcuminide biosynthesis pathway is unique to this plant, right? So we showed this pathway. It's the gene structure of the genes, and also like what how this pathway is evolved. Then we published the first genome sequence of aloe vera, which was also published this year, and this is the largest endosperm sequence till date. It is 5 times bigger than the human genome, so it is a very, very big project, right? So we had to do a lot of sequencing here, and this is also the largest genome sequence so far using the X4 nanopore technology, right? And it provided a lot of insights about how this genome survives in difficult drought and stress conditions, right? And of course, both turmeric and aloe vera are very important genomes medicinally and commercially, right? Then we recently published the first genome sequence of Aloe, which is widely used in COVID, right? And so basically we looked at the genes that are responsible for the various antibacterial, antifungal, anti-cancerous activities, and also looked at the genomic basis of the origin of these medicinal properties. Okay. Then recently, last week, we also published the genome sequence of the national tree of India, that is banyan tree and the peepal tree. And again, we use the 10x and nanopore reads. So I'm not going to details of these plants. These are already available papers, and I just wanted to show a glimpse of these so that what kind of things are going on here, and right. Okay, and at the end, I would thank all my students who contributed to the work that we did, right? Which I presented today, my collaborators and the funding agencies, and ISOPOPAL for all the support that they provided. I'll just look at the chat. And you have to sequence my favorite plant genome also, please. Okay. And Gitanjali, so what is your favorite genome?
Sorry, Vineet, I'm butting into your talk, but you have to sequence lantana camera for me. I'm going to give a talk about it and all of our work on biodiversity informatics today. But sequence genome ka nahi hai hamare paas. Wo tumko dena padega.
Yeah,
but I'll catch you later.
Yeah, sure.
Great to hear you.
Yes. Yes, I'm open to questions. Please, I'm sorry for the exceeding the time. Yes.